Well, good morning, everybody, also the audience, welcome. Hereby I open officially this academic ceremony in which Mr. Ben Rombaut will defend his academic thesis entitled Out of Control Microglia Cause Cognitive Deficits to Excessive Synaptic Elimination in Alzheimer's Disease. Mr. Candidate, may I invite you to present a summary of your studies and the conclusions of your thesis in the next 15 minutes. I give you the word.
Members of the jury, promotion team, family, and friends, thank you all for joining me on this day where I will explain my PhD project about out-of-control microglia in Alzheimer's disease. As modern medicine keeps improving, the global population continues to grow older. Average life expectancy in Belgium and the Netherlands is 81.7 years old, which is 10 years longer than 50 years ago. This is a great accomplishment, but as any elderly person will tell you, this number is only important as long as you're healthy. Sadly, for about 1 in 3 aging people, their golden years are disturbed by dementia. Alzheimer's disease is the most common form of dementia, as 55 million people worldwide suffer from it. People suffering from Alzheimer's disease have trouble following conversations, issues regarding time and place, difficulties performing everyday tasks, mood swings, as well as problems with memory. Today, I will be talking mostly about memory, as it is the most well-known symptom. Memory, as we know it, is created by communication of brain cells. These brain cells, called neurons, are the functional cells of our central nervous system. Now, on this beautiful day that we're all together here, we are all making a memory. We all see and hear what is happening here today, and all this information is being processed by our neurons. The neurons pass along this information to one another where their branches meet and make sure that the signal gets where they need to be. A couple of minutes ago, I walked in here, and this is what happens in our brains. The signal gets passed along from one neuron to another up until its endpoint. If we zoom in a little closer on these branches, we see highly specialized contact points called synapses. These synapses are what make sure that the signal travels from one neuron to another, and they are made or strengthened when we need to store any of the information we process, like pages in our diary. So every time we learn or remember something, one of these pathways is made through these synapses. You can compare it to writing your fondest memories in pages of your diary. Looking around this beautiful room today, there is a lot of information to be processed. For instance, by now you will have probably noticed the outfit of the person sitting next to you. This information, to me at least, would not be worthwhile remembering for a long time. So now that we understand memorizing, I'll explain something that is equally as important: forgetting. If we wouldn't forget anything, there would be an information overload in our brains, and remembering would be terribly inefficient. So we know that memories are stored in pathways created by synapses. So to be able to forget, these synapses need to be removed, just as taking away pages from our diary. In a healthy person, there is a constant balance between creating memories and forgetting them. There is a certain number of synapses in the healthy brain at any time, with constant creation of new synapses and removal of old synapses. However, in the brain of Alzheimer's patients, we see significantly less synapses. This means that too many of these synapses are being removed and memories are forgotten. Overseeing these processes are a certain cell called microglia. Now, microglia are the immune cells of the brain, which make sure that all these processes happen as they should and that no outside infection can run rampant in the brain. Microglia are equipped with a certain number of scissors in the healthy brain, which gives them the ability to cut away these synapses and eat and remove them. However, in the Alzheimer's disease brain, this balance is disrupted because we see that they are equipped with too many scissors, which leads to excessive loss of synapses and the loss of memory as we know it. Now, what drives this imbalance is a certain protein called phosphodiesterases, or PDEs for short. Now, these PDEs are a certain type of protein, an enzyme, that can alter microglial functioning. And when there are too many PDEs present, these microglia become out of control, running rampant, and destroying too many synapses, which leads to the memory loss in Alzheimer's disease as we know it. So, to quickly recap, in the healthy brain, we have a certain number of phosphodiesterases, or PDEs, and this leads to a baseline number of scissors, which causes microglia to do their task in removing synapses as pages in a diary to maintain a healthy synaptic network. However, when there are too many PDEs, as in the Alzheimer's disease brain, this baseline number of scissors heightens, which leads to excessive removal of synapses as pages in our diary. Now, the attentive listener will by now have figured out that scissors cause problems to papers. So what beats scissors? That's right, it's rock. And our rock, in our case, is a certain drug that targets these phosphodiesterases. So, in theory, we would hope to use a drug targeting phosphodiesterases, or PDEs, to lower the amount of scissors in Alzheimer's disease, causing the microglia to go back to their resting state, and thus preserving the synapses from getting lost. Now, how did we do this? Well, first of all, we isolated some of these synapses, we colored them red, and then we gave them to our microglial cells in the lab. Now, microglial cells are equipped with a certain number of scissors, so they will use these to cut up the synapses and to eat them. Now, the red color of the synapses that we gave to them will then be visible in the microglia. So for short, the more synapses the microglia have eaten, the more red they will become. And this red signal is something we can measure in our lab. So if they have eaten a lot of synapses, we would expect a lot of red signal, and we did indeed see this in our lab. Now on to the main question. Can our inhibitor counteract this? So first of all, when we gave the drug, we would expect the microglia to be equipped with less scissors to eat these synapses. So if we then give them these red-colored synapses, we would expect them to cut them up and eat them, but to a lesser extent, which would translate in less red signal emitted. And did we measure this in the lab? We did, in fact, measure this. So by pre-treating with our drug that targets phosphodiesterases, or PDEs, we could let them eat less synapses. Then we went on to see if the drug did indeed help memory. We tested this using mice that have some genetic flaw that causes them to develop Alzheimer's disease. Now, these mice, when they are young, their brains behave just like a normal, healthy mouse. Namely, microglia are equipped with a certain number of scissors, which leads them to be able to create new memories and forget them. However, when they become teenagers, these microglia become out of control, causing them to remove too many synapses and to lose memories when they are older. So, in this setup, we believed that treating these animals before these microglia go out of control could lead them to preserve these memories. Now, you might ask, how exactly do you check the memory of a mouse? Well, simply put, we use the object location task, which is an arena that uses two identical objects. During 4 minutes, we let the mouse freely explore the arena to familiarize itself with the objects, and then remove the mouse. After a certain time interval, we move one of the two identical objects, and we reintroduce the mouse for 4 minutes. If the mouse has a working intact memory, it will see that the new object is moved, and thus see it as a new object. Now, mice are inherently very curious animals, so they will start exploring the new object more, and we can measure this time. So if we measure all these times, we analyze it using a certain discrimination index, or D2 value. Now, simply put, if the mouse explores the two objects the same amount of time, the D2 value should be close to or equal to 0. And if the mouse has an intact working memory and spends more time at the new object, this D2 value will be significantly higher than 0. So on to the results. First of all, we tested brothers and sisters of the mice that did not have the genetic flaw and are thus considered healthy controls. So if everything is right, they should have intact memory, and we did indeed measure this. So these mice had intact memory, they recognized the new object, causing them to spend more time there, which we measured as a higher time and a higher D2 value. So their siblings who did develop Alzheimer's disease were next, and they should have an affected memory and a lower score. And indeed, we did measure a very low score, leading us to believe that their memory is affected. But then the big question remains, of course, does pre-treatment with our drug increase memory or preserve it? And we did indeed measure higher D2 scores, which leads us to believe that their memory is intact. So by pre-treating with a drug before memory loss occurs, we can preserve memory in these animals. But the question remains, is this due to the number of synapses? So afterwards, we tested the same mice in their brains to check the synaptic network. And indeed, in the healthy controls, we found a very dense synaptic network. In a certain field of view, there were at least 500 synapses. So it would stand to reason that in the Alzheimer's animals, we would find less synapses. And indeed, in a similar field of view, we found around 200 synapses, which is a way less dense synaptic network. But again, the question remains, does pre-treatment with a drug lead to synapse sparing and thus a more dense network? Yes, it did. It restored the synaptic network almost to baseline levels in healthy controls, which was very good news, which meant that in these animals, by pre-treating with the drug targeting PDEs, we could preserve memory by protecting synapses. So to conclude, some of the take-home messages I would like to leave you with today are these. In normal brain functioning, microglia are equipped with a certain number of scissors to remove synapses, to maintain a healthy balance of memorizing and forgetting. This keeps the memory intact. However, in Alzheimer's disease, these microglia go out of control, causing them to remove too many synapses, leading to memory problems down the line. In these mice, we tried to prevent this going out of control by the microglia by targeting PDEs, and by doing so before they cause their damage, we could prevent the loss of memory in these animals by sparing their synapses. Now, most of the work I talked about today was enveloped in chapter 3 of my thesis, but that's not the only thing I did during this 5 years. Namely, in chapter 2, I bundled my literature study into a review discussing all the basic principles of targeting PDEs in different cell types to have a positive influence on synapses. In chapter 4, I talk about another gene that regulates PDE4 called DISC1. This story was not as straightforward as the one I told you today, but I believe it's still valuable information for a fundamental understanding of the disease. Finally, in chapter 5, I discuss all of the gathered data more in depth, and I reflect on what they could mean for understanding the disease as a whole. And with that, I conclude my presentation for today. I give the word back to the prorector and welcome any questions or remarks.
Thank you very much, Mr. Candidate, for your clear presentation, well illustrated. It was indeed formulated in the way we like to see it here at Maastricht University, a typically presentation for a lay public, and I think I compliment you with this one.
Thank you.
Before we proceed, I also would like to the audience introduce the promotion team. So the team who brought you in the last 5 years, that you can stand here this morning and defend your thesis. And I would like to mention that the promotion team is sitting opposite of me for the audience. And I start with Dr. Wens. She is involved in the Department of Neuroscience at Maastricht University. Then we have Professor Bronne from the Department of also Neuroscience, but now Maastricht University, the different one. Then we have Professor van den Hove, who is involved in the Department of Neuroepigenetics at Maastricht University. And finally, the first promoter is Professor van Mierlo, and he is involved in the field of glia biology at Maastricht University. That's the team who brought you in the last year. I received from the first promoter a list of opponents which I'll follow. And for the audience, I would like to mention we have 4 members here online, 4 members here on site, and we have 2 members on site, online. They are appearing on the screen later, or they're already there, and they will afterwards appear on the side screen. And we will start the opposition with the chairman of the assessment committee, which is Professor Visser. Professor Visser is professor in molecular epidemiology of Alzheimer's disease at Maastricht University, and I'd like to give him the opportunity first to discuss.
Thank you. Dear candidate, I would like to exchange some ideas with you. But first, to congratulate you with a nice book, thesis, and also congratulations for your supervisor teams. One of the central themes is the protein disc C1, and it has been discovered in schizophrenia. I wondered whether there's a shared mechanism between Alzheimer's disease, in which disc 1 plays a role as well, with schizophrenia, so there's pruning in schizophrenia as well.
So, highly esteemed opponent, thank you for your kind words and your insightful question. If I understand it correctly, you're asking if there is a mechanistic link between schizophrenia and Alzheimer's in the case of disc 1.
Yeah, so whether this one has a common activity in schizophrenia and in Alzheimer's disease.
Yes, so disc 1 as a protein is very diverse in its functions. It has many interacting partners in many different cell types, but I will limit myself to microglia in this case. I believe that there are some common pathways. The DISC1 protein on its own has been linked to cytoskeleton changes in actin. I believe mainly for phagocytosis, this is of course very important, the cytoskeleton. And I think there might be some causal mechanistic similarities, but I believe in schizophrenia it is more the opposite route, so that there's too many synapses. So I believe there it's more a story about maintenance rather than removal. And also in schizophrenia, the timeline is way different. So in schizophrenic patients usually develop symptoms around adolescence. But I believe mechanistically, the effect of DISC1 in microglia on synapses, there could be some similarities, yes.
Okay, thank you. Then about the pruning, so you mentioned that the microglia are out of control. So what's the role of amyloid pathology in this?
The amyloid pathology in the animals that I tested?
Yes, well, in general with the pruning. So it's not only pruning because there are microglia, but there's pruning because there's Alzheimer's disease as well.
Yes, of course. Well, there is indeed a difference between the physiological pruning that is necessary for maintenance and the damaging pruning. I believe it's not very clear what the role of amyloid beta plaques in this is. I think there are two trains of thought, one being that the damage to synapses caused around amyloid beta plaques being more of a collateral damage nature, or whether that these synapses are actually tagged by the amyloid beta for removal. I kind of tried to look at it independently of amyloid beta. But I believe, yeah, it cannot be disregarded, of course. There is a very important role for amyloid beta in this.
Yeah, so I think without amyloid pathology, there's no aberrant pruning whatsoever, I guess. So you briefly mentioned plaques. So microglia like to get rid of the plaques as well. Do you think there's a role of DISC in that and PDE as well? So it was not the focus of your thesis, but that's one of the other roles of the microglia.
You're asking if there is a role for PDEs in microglia and plaque removal?
Yes.
I believe there are many different roles for many different PDEs in microglia. I think that the role of PDE in microglia does extend further than phagocytosis of just synapses. Of course, in an early on stage, before they are plaques, when the amyloid beta is still in its fibrillic or oligomeric stages, I believe that there is active phagocytosis by microglia. But I believe that when it's in the plaque stages, that microglia kind of converge around it, but do not phagocytose, rather than stimulating an inflammatory microenvironment. And PDE4B is, of course, also very well known for its role in this inflammatory stimulus.
Yes, okay, thank you. I think final question. So the results of your experiments are a bit modest. So which other target would you like to investigate in order to avoid pruning of synapses?
I think there are many interesting targets for pruning of synapses. The complement cascade, for one, would be interesting to look into, because this research did not take into account many of the eat me or don't eat me signals that synapses could express. So I believe including these proteins like phosphatidylserine or the complement cascade would also be very interesting.
And one major genetic risk gene for Alzheimer's disease?
ApoE, for example, or TREM2.
Yes, TREM2, yeah. So I think for treatment development, there's a strong focus in all big pharma on TREM2.
Yes.
And maybe that has more effect on the reduction of the pruning, but we have to see.
Yes.
Okay, thank you.
Yep.
I give the word back to the pro-rector.
Thank you very much. Then the opposition will be continued by Professor De Wachter. Professor De Wachter is head of the Department of Neuroscience and Neurodegeneration, in particular in Alzheimer's disease, from Maastricht University. I'd like to welcome her here in Maastricht, and I'd like to give her the word.
Thank you, Pro-Rector. Dear candidate, I want to start with congratulating you with this very extensive work. You've been doing a lot of experiments in vitro, in vivo, in humans, and on different topics, Alzheimer's disease, and then this one related. So congratulations also with the presentation today and with the booklet. PDA is a very interesting target. There are multiple forms, and I wonder why did you choose PDE4B while expression in microglia is low, and how does it relate with PDE4D, which is also investigated in the context of AD?
Highly esteemed opponent, thank you for your kind words and your insightful question. The choice for PDE4B was mainly related to its expression. I agree that in humans it is expressed quite lowly, and there are other targets like PDE4D, as you mentioned, or PDE3B, which are also highly expressed. But I believe the role of PDE4B in microglia cannot be disregarded. It was already well established, mainly on inflammation and such. But the choice for PDE4B was kind of evident as the role of PDE4D seemed more neuronal to us. Well, I also did some work on PDE4D in microglia, confirming their differences in expression pattern, mainly in the role of DISC1. So for DISC1, we opted to include it because it was a known interactor. But for our Alzheimer's story, we opted to chase PDE4B, mainly to take it out of its inflammatory role and into a new pathway we thought was interesting.
Okay, thank you. Then I would like to ask a more technical question on figure 2 of your chapter 3. It's the effect of your inhibitor on the object location task. So can you indicate what kind of tests you have used and whether also you have considered other tests and what would they learn us, or the advantage of this test and other tests?
So you are referring mainly to panel A, the object location task?
Yeah.
So we opted for the object location task because it's quite standard in our lab. For other tests, we could also consider the novel object recognition, but the object location is a staple that we always do, just as the Y-maze. And considering the results that we had with this test, we opted to remain here. We used a one-sample t-test, and I believe that, this data recapitulated nicely what we wanted to see. It's always an option to keep doing behavioral tests, of course, but you don't want to overdo it with behavioral tests, I think.
Could you also use single ANOVA for the statistical analysis?
You could use an ANOVA test to compare between groups, and this is usually what you would do. But for our case, we used the one-sample t-test mainly to establish intact memory or affected memory. Okay,
thank you. I would like to continue a little bit on the question of the previous jury member on the phagocytosis and on the link with A-beta. You find effects on soluble A-beta, and you see effects there. And how would you see your effect on phagocytosis of A-beta and phagocytosis of synapses? And do you think those mechanisms are alike or are a little bit different?
So obviously the broader mechanism of phagocytosis is alike, but I believe that these two operate on very different pathways. So as the previous opponent mentioned, TREM2 is very important in certain phagocytic pathways. There's also toll-like receptor 4 that is important for bacterial phagocytosis. So I think they're wildly different, of course. I think that in response to amyloid beta, also the expression pattern in microglia differs completely. So yeah, this is something I haven't really taken into account, just as the eat me, don't eat me signals also infer differences in the microglia. So too will the different forms of A beta, I believe.
I have a last question. I liked your comparison with the rock, sheet, and scissor. Is there a sheet for your rock? I mean...
Yes, I kind of expected this question. That's why I didn't include the circular arrow. So no, I don't. Well, I think that the synapses also influence the microglia, of course, but I don't think the synapses will.
I'm thinking about side effects. Do we have to consider side effects or something like that?
Of course, as any treatment, this one will also bring its fair share of side effects. For one, the treatment is not specific to microglia, so neurons, endothelial cells. Yeah, this can of course not be disregarded.
Thank you.
Thank you. Then the opposition will be continued by Dr. Aguiar Alpizar. He was a member of the assessment committee, and he is affiliated with the Department of Microglia Physiology at the Catholic University in Leuven, and I would like to welcome here that he likes to join this morning, and I'd like to give you the word.
Yeah, thank you, Bruno. Thank you, Mr. Candidate, for your clear presentation. I also want to extend my congratulations and for the work for your presentation. And of course, I have some questions because it's a very nice book, and very nice work, and it just triggers many, many questions. Briefly, in chapter 1, you mentioned that microglia exist in or can be found in 3 states, different activation states, and this is mainly characterized, as we have seen, by a different gene signature. So regarding PDEs, what is the gene signature, and how do you think this changes it, or if you know that this is differentially expressed, especially in the DAM phenotype, and how would this affect then your treatment?
So, esteemed opponent, thank you for the kind words and your interesting question. I believe you're inferring to the expression patterns of different PDEs in these different signature microglia. Yeah, so I believe there's of course many states of microglia, of which I kind of highlighted the disease associated as being of interest. I think the disease associated microglia as a whole have varying expression patterns of PDEs, and I think this is within this group even more fractured based on their spatiotemporal positioning. So if they are close to plaques or close to degenerating neurons, I believe that PDEs will be heightened in response. Mainly, if I speak mainly about PDE4B as an inflammatory response, that's obvious, but also changes in PDE4D and PDE3B have already been established. So I think, trying to target one single PDE in this vastly different network of spatiotemporal differences is always finding a needle in a haystack, I suppose. So in that regard, my setup is not perfect. But I do believe that targeting PDE4B can be of interest in multiple of these signatures of microglia.
Okay. Thanks. Also regarding the limitations, and then I'll come back on a question from Dr. De Wachter, Professor De Wachter. Regarding the limitations, so most of the experiments that you show on microglia morphology are in vitro experiments, where, of course, there is no recapitulation to a real 3D environment of a microglia. Brain organoids may be a solution for this. So what's your take on the use of brain organoids for this type of experiment regarding media stimulation, ratio of cells are included?
So I agree. Of course, in vitro microglia behave wildly different than in vivo microglia, so this should also always be taken with a grain of salt. I believe that the use of organoids can be very valuable information in this, but then, of course, introducing the isolated synapses is also not perfect. So there's always some wiggle room you have to work with. But I believe your suggestion of brain organoids could be very interesting, also just to see if you only knock out one gene in the microglia and introduce them into an otherwise normal environment of neurons. Yeah, that would be highly valuable information, I believe.
Yeah. So regarding the access of the drug, for instance, in this case, how would you think it would be different if you would do it in a real individual and not and in brain organoids, because there are different barriers and different other metabolic pathways that need to be that are involved in real people that are not in brain organoids.
Yeah, of course, the passage to the blood-brain barrier and the metabolization of the drugs is wildly different. So we, of course, try to approximate these doses in our experiments. But yeah, that would be in a late stage, which clinical trials should yield more information about. But I believe I'm not quite there yet.
Yeah,
right?
yeah. Okay, if I may, last question regarding the side effects of the. Yeah, okay, yeah.
There is room for more questions if you want. Don't hesitate.
Regarding the side effects, so that were touched upon already, I'm more concerned on the side effects on the microglia that you don't want to target. Because also connected to the first question, there are microglia that don't need to be targeted for this reason. And how do you see the balance on the healthy microglia, and what will be the side effects also on the regular homeostatic function of microglia if you're targeting PDE?
Yeah, of course, there's also a large subset of microglia that are there to help or do their jobs, I suppose. And you wouldn't want to target these. That is absolutely correct. In this, I believe that my experimental setup of just general PDE inhibition everywhere is, of course, lacking. I think that more targeted deliveries towards the more disease-associated or the more plaque-associated microglia would be what you would aim for. So yeah, I agree that the side effects on the microglia themselves are less than ideal. But using PDE inhibition, there's lots of compensatory mechanisms, of course, which only makes it more difficult. But I agree that this modality of treatment should take that into account.
Okay, thank you, Director. I'm finished with my questions. Thank you. Yeah.
Okay, thank you very much. Then the opposition will be continued by Dr. Sierksma. She was also a member of your assessment committee, and she is involved in the Department of Neurodegeneration, also at the KU Leuven in Belgium. And I'd like to thank her that she joins this morning. And I'd like to give you the word.
Thank you. Congratulations, dear candidate, with the nice work. You did a lot of work, indeed, a broad scope of experiments. Also, congratulations to the promotion promotion team. Sorry. So I would like to exchange some thoughts. I was intrigued by statement number 4, so I'm not sure if one of your paranymphs could read it out loud. We're all on the same page.
Statement 4. Clearly, aberrant microglia functioning starts happening long before the decline in cognitive capacities. As such, it should be targeted as early as possible.
Brilliant, well done. Okay, so I was wondering, what do you consider aberrant microglia functioning? What is your definition of aberrant microglia functioning?
Esteemed opponent, thank you for your kind words and your insightful question. So what I would consider aberrant microglial functioning is, of course, the very broad term that I used here, the out of control. I believe that there is a physiological level of pruning that is, of course, necessary. But when this happens in excess, or as we previously discussed, as there is collateral damage to synapses when they try to clear plaques or do other homeostatic influences, I think that is when it crosses the threshold of being aberrant.
Okay, and do you think that this could be, is this something that's always there, or can this wax and wane?
I think there is definitely an ebb and flow to it. I think based on the microenvironment and what is happening there, that microglia are equipped to handle it, so they are very sensitive to what happens in their microenvironment and they react to it, which is of course necessary. But I think when their efforts are futile and they get in a kind of vicious circle of reactivity, that is when the aberrant functioning happens and that the damage occurs.
Okay, and indeed also a bit alluding to the question of my neighbor, how do you see this then for targeting strategies? Like how would you go about this? What would be your ideal scenario of how to treat then these aberrant microglia?
So the ideal scenario, I believe, would be very reactive as well. So we know of the different signatures and states that they can travel between. And ideally, you would want a very limited response. So when you see that too many microglia are traveling from, for instance, homeostatic to reactive to disease-associated, that you want something to drive them back to the reactive or possibly even the homeostatic state to do more good than harm. As to how you would attain this goal, that for now eludes me still.
Okay, good. Okay, I'll shift gears a bit. So I had some questions regarding chapter 4, figure 10, where you do these phagocytosis experiments, where I think you either give a continuous stimulus or a 40-minute kind of pulse chase experiment where you give the stimulus and then you take it away again. At least that's how I understood it. So I was wondering about this because what would be your definition of phagocytosis?
So I believe you're referring to the in vitro eating of synaptosomes?
Yes, sorry. Yeah, and indeed, the pH Roto experiments.
Yes,
yeah.
So in the pH Roto experiments, what we actually measure is when they get broken down. So when the phagosome and the lysosome lyse and then break down through acidification what they ate, that is what we measure. Now, I agree that this adds a level of complexity to it. So are you actually measuring what is being taken up or what is being efficiently removed? Because if something is being taken up but not efficiently removed, you lose this signal in this setup. So that is something we kind of struggled with in this experimental setup as well, but that is what made us land on the 40-minute stimulation, taking it away and then measuring, because there was also an experiment where I did continuous stimulation and you just saw the data kind of bleed together. But there's also some experiments performed by a colleague where we used the dye-I label synaptosomes, so which are inherently fluorescent. There I think you have a measure of exactly what is taken up. But then of course, if that gets broken down, then you lose your signal. So there's no perfect modality which includes both of them, but I think comparing the two of them as well as possible should, be able to conclude something about that.
Yeah. And do you know of other systems to measure lysosomal efficiency or acidification of the lysosome? Are there additional experiments you could think of that you could you could do to untangle indeed what's the difference in phagocytosis? Is it uptake? Is it breakdown? Can you think of other experiments that you could do?
Well, I imagine that the breakdown products have to be shuttled off anyhow. So I think measuring any pathways that use this shuttling off or forming of synaptic vesicles might be of interest to figure out the rate of clearance as well. But I'm not well versed in those experiments.
Okay. And do you know whether cyclic AMP or DISC1 could also influence the endosomal lysosomal system?
I believe cyclic AMP can involve the endosomal system, but I have to admit to which degree, I'm not sure.
Okay. All right, I'm shifting gears again a bit. So this was on, I think in both chapters you describe your bone marrow transplantation experiments. So I was wondering whether you can explain to me whether there's a difference between bone marrow-derived macrophages and microglia, and why do you think this treatment strategy is a good option?
So yes, these cells are wildly different. One paper by Jonathan Kipnis, of which we kind of based our procedure, talks about the wildly different transcription factors and just transcriptome between these two cells. So actually you should call these newly introduced cells microglia-like cells because they are completely different, of course. Purely through origin, the microglia are derived from the yolk sac and the macrophages are not. And they have more of a macrophage-like phenotype. So I agree, they are wildly different. And as such, as a treatment, I don't think this is ideal. Far from it, even. It's a very invasive procedure, and it's very prone to failures and errors, as you might probably have read. But I think, just as with the question of the organoids, the main goal of it was really to introduce these aberrant microglia to an otherwise normal environment, and in that case, I believe we had a chimerism of above 80%, which is admirable for the amount of information we wanted to get out of it.
And can you think of alternative strategies how you would, how you could answer the question that you have, right? To what extent microglia expressing specific proteins or not expressing certain proteins, how that could affect the pruning and the homeostasis of the system?
So I believe, yes, probably in the time window that we described that the pruning does happen, I think you could do some signal cell analysis to figure out exactly which signature your microglia has, and if it is in this possibly hyperactivated state, that you could figure out which proteins are involved in the synaptic clearance.
But could there be other ways, instead of this bone marrow transplantation experiment, to do how to knock out disc 1, for example, in microglia and see what the effect is?
Yeah, for instance, a Krelox system, but we opted not to do it for the disc 1 because it is a kind of complicated combination of mutations, which would take us way too far. But there are other models which you could use to simulate the knockdown of disc 1, yes.
Okay. All right, I give the floor back to the prorector.
Thank you very much. Then the opposition will be continued online. We move now to Professor Ponsarts. He is Professor of Immunology at the University of Antwerp. And I would like to welcome him and I'd like to give him the word. Professor Ponsarts, please.
Okay, thank you for the introduction. Dear candidate, can you hear me well?
I can hear you.
Okay. I think you gave a very, very nice presentation, a general presentation for a layman's public. But I don't know if it's possible to go back to your slides. Because you made this beautiful claim that with the therapy you have foreseen, that you almost have a black and white result on having cognition back to normal and having the synapse connections coming back to normal after treatment. So, and in a little bit, I have it quite difficult to look at this result back to the results in your thesis, because to me, they are not so black and white. So maybe if on the graphs that you have shown, can you actually just visually tell me what is the type of variation?
Thank you for your kind words and your insightful question. I agree that this, this graph is of course highly simplified, and as you stated and nicely noticed, it is indeed very black and white to illustrate my point. And I agree that the data in the thesis itself is not as black and white. Sadly, working with live animals has some drawbacks, variation being one of them. But I believe that by the analysis that I did with the one sample t-test, significantly different from 0, as I try to explain in layman's terms, the D2 value is of utmost importance. We're
not going on the statistics, but if you would look on this graph, just we all know it's not going to be black and white, a treatment for Alzheimer's disease. Where would you draw the line taking into consideration the data you have in your thesis, because this is a graph based on whether or not the effect was there. So how efficient would you assume the therapy to be?
That is a difficult question to answer. Of course, you would hope.
I know.
Hope 100% of the time it works every time, but yeah, I believe this data cannot claim that we have found the end-all, be-all therapy for Alzheimer's. Far from it. But I think mechanistically, the point that I'm trying to make or have come across is that early preventative treatment has profound effects later on. Just the time window of treatment was when they were stopped when they were 4 months, and the measuring of memory was when they were 9 to 10 months. So by treating a half year earlier, you still see these marked results half year later. Is, I believe, the main point I'm trying to make. The fact that I made it so black and white in my presentation might just be, yeah, making life.
Okay, we all understand. Yeah. I want to change a little bit, and we already had a question on this on your microglia replacement experiments. And I've worked with that mouse model for many, many years. And we also made iPSC from this mouse model, and we made from those iPSC microglia and macrophages. So, and you already answered correctly that we should talk about microglia-like cells instead of microglia after the replacement. So I would like to know from your impression, when you did the analysis, when you looked under the microscope, the level of EGFP expression that you saw after the bone marrow transplantation in your microglia-like cells, is it as bright as the EGFP in the original mouse or not?
So if I understand it correctly, you're asking that whether the brightness of the GFP in the introduced microglia-like cells is comparable to that of the donor mice on its own?
Yes, comparable to the microglia of the donor mice.
I have to admit, I did not check for this, but I would hope so. Of course, this is a young man's dream. A young man's dream. But for our experiment, we just needed them to be green. We of course understand the implications of tagging the CXCR1 receptor with GFP, which brings along loads of problems on its own, which is why we opted for the heterozygous knock-in. But I think that's
I would suggest for your promoters to look back at the original data, because normally the EGFP in the microglia in this mouse model is much brighter than the ones in the bone marrow. So you can compare the level and see whether you are like a bone marrow derivative or a true microglia. So maybe something to check what you can still do. And then maybe a last question, if I still have time. And we touched already upon organoids and the usefulness of organoids. And you may or may not know, we culture a lot of organoids in the lab, also from this mouse model, with and without microglia. And during your presentation, I quickly looked back at my RNA-seq data on those organoids, and especially looking at PDE. So I want to have a little bit of a thought of you from, because my personal interest is not in PDEs, but I just quickly looked at it. So if I have mouse organoids having neurons, astrocytes, and microglia, and I've stimulated them with LPS, so basically creating a pro-inflammatory environment, maybe what compares to your model, having overactivated microglia. What I see in this condition is that PDE4 goes significantly up and PDE6 goes significantly down. Would that make any sense to you?
Well, it would make just as much sense as the whole of science does. As in, I find it interesting. It does make sense, of course, that PDE4 goes up. Now, stimulation with LPS, I believe, more coincides with bacterial infection than So yes, in that case, PDE4 going up makes absolute sense. It is established in inflammation. And the fact that PDE6 goes down, yeah, that's very interesting. As I scream from the rooftops, there is compensation in PDEs, and it's always more difficult than it seems. So yeah, if you want to start a follow-up project about PDE6.
So if PDE4 goes up, okay, I'm almost finished. So if PDE4 goes up, okay. So if PDE4 goes up, can I correlate it with loss of synaptic function?
Well,
in an organoid?
That is a tough conclusion to make. I don't think saying PDE4 bad, so it going up, is automatically bad. But I believe, yeah, it would be interesting to look at. I think if PDE4 goes up in your microglia, obviously they are stimulated to react. But I believe this is probably an acute effect of the LPS. Whether this does incur long-term effects on synapses, that remains to be seen. I think the acute effect of LPS is, of course, multiplying the expression of PDE dramatically, but whether this is sustained long enough to have marked effects on synapses, yeah, that would be interesting to see in organoids.
Okay, thank you very much.
Okay, thank you very much. Then the opposition will be continued, also online, by Dr. Havekes, and he is involved in the Department of Neuroscience of Memory and Sleep at the University of Groningen, and I'd like to welcome him, that he joins this morning here at this thesis defense, and I'd like to give him the word. Professor Havekes, I think he will appear now on the screen.
Thank you, Pro-Rector. Dear candidates, can you hear me well? I don't hear you,
but I can hear you, yes.
Thank you. Thank you. Well, first of all, congratulations also on the completion of your thesis. It was very nice to read, and I was particularly impressed with the large number of approaches and techniques that you applied for your studies. And I hereby would also like to congratulate your promoters and co-promoters with the completion of this elegant piece of work. Finally, a nice compliment also on the nice layman's talk that you gave prior to this defense. The first thing I would like to talk about, and it has been brought up already a few times, is the object location memory data from figure 2 in chapter 3, where you stated in addressing the question from Professor De Wachter that you used a one sample t-test, but then in chapter 4, figure 9b, you also have object location memory training, and there you chose a different statistical method to analyze the data. Could you elaborate a bit on decisions for one experiment to take one statistical approach and then for the other set a different one?
So, esteemed opponent, thank you for your kind words and your very detailed, insightful question. Of course, the similarities between the two is that I always use the one sample t-test to infer loss of memory or intact memory. And to be perfectly honest, the fact that I use a comparison between groups is just because it was nice in the figure in chapter 4, because there is marked difference. And well, I also tested it in the figure in chapter 3, of course, but drawing lines that say not significant just doesn't do it for me.
Okay, and so that means that sort of it seems that, let's say from a positive side, that the resolution in this task, at least for the study as you've done it in chapter 3, might have been not optimal. Would there, if you would be able to, let's say, do the study again, would you try to change something in the design of the behavioral test to maybe increase the resolution for these studies?
Yeah, looking back at experiments, there's always something you would hope to try different or improve. I think, for one, improving the sample size, which is probably the dream of every scientist, infinite animals doing exactly what you want. But other than that, yeah, I think the treatment modality, as has been discussed by many opponents before, would also, there's room for improvement. So I think if we could find a way to treat just the microglia that we want, this could get rid of some confounding factors that possibly have an effect on behavior, because the effect of PDE4 inhibition on behavior is, of course, well established. But I think the strength in it does still lie in the timing of treatment, where, as opposed to the timing of the memory tests.
Okay, so you wouldn't change any parameter of the test itself, if I understand it correctly.
Sorry, I wouldn't change any?
You wouldn't change any of the parameters of the behavioral task itself?
Well, the behavioral tasks in our lab are pretty standardized. I would maybe try a different time interval in between. This time interval was the one that seemed to work for this test. But ideally, you would want a time interval of 3 hours is usually where we see it. But yeah, of course, this experiment had many of its own little quirks that kind of changed these parameters as we.
I can agree that definitely, the behavioral studies are not easy to do. Now, let's continue with these behavioral studies, but then in chapter 4. So here you describe a series of phenotypes in the disc mutant mice, and in general, it seems that they have reduced activity. Now, to what extent do you think that this reduced activity also plays a role in the data that you show in figures 7 and figure 8?
I believe you're referring to the data of the open field, where they show reduced locomotion and activity. And if this could be a confounding factor in the memory tasks. Well, we did...
Now, particularly in the suppressed instinctive behavior, for example.
Well, we did kind of test for this, but the suppressed instinctive behavior mostly looks at the marble burying and the nestlet shredding, I presume. Well, as we stated in the marble burying, it's only 30 minutes. And if you take a look at the mice doing it, they do seem not less active, just less engaged sometimes, which is of course not what you want in a behavioral test. But we were strengthened by the fact that the wild type animals at least performed as they should at exactly 10 marbles. And for the nestlet score, yeah, this is a test that we performed over 24 hours. Of course, if they are less active, this is also going to affect this. But for these tests, they were solitary housed and left to their own devices. So yeah, it could be a confounding factor, but I believe these were concerns that were raised while I was doing the tests. So every single test that was added on was for one of these, to address one of these specific reasons.
Great, thank you for that. Now, on a similar note, I would like to discuss the object location memory data in figure 9, so figure 9b. There you show a nice and robust effect. But also here, as you said already, if they're less engaged, it might actually affect their behavior during the training. So my question is here, have you also analyzed the training data? And I would be very curious to hear what you found there.
Yes, I have not included it in the supplemental data, which, yeah, now that you mention it, it might be an oversight, but we did indeed check these times during training. So exploration times over genotypes were not significantly different. They did not prefer one object over the other, and they did not prefer the left object more over the right. So all of the parameters and training modalities that were necessary to check were checked, and there were no significant differences found.
Wonderful, glad to hear that. Now, for the sake of time, I would like to move on to a much more general question. It seems that based on your thesis, that the PDE4B family seems to be sort of a molecular constraint on cognition, and that we should inhibit it. Do you agree on that? If it does not, then I can.
Sorry, you're asking if PDE4B is directly a modality to treat cognition?
No, if the inhibition of PD4B is sort of something that we should do because it is a molecular constraint on cognition. Now, that's not really the question, that's my statement. My point is, if PD4B is such a bad player in memory, why would it, across evolution, not have been already sort of silenced?
Yeah, we get this question a lot from a professor in Hasselt who always says, if PDEs is so bad, then just put the inhibitors in the drinking water and be done with it, which of course would be very nice if it were the case. But yeah, this of course deserves some nuance. I don't think it's always the case of PDE bad, inhibited, and good results will follow. I think this should be looked at in a case-by-case manner, as with all therapy strategies for Alzheimer. I think there is no silver bullet, and obviously PDE inhibition will not be that silver bullet either.
But would you have any idea about what the good processes are that the PDE4B might play a role in?
You are allowed to answer, but you don't have to answer. That's the law over here. So that's the choice is to you.
I will answer the question to humor Professor Havekes, because the answer is no, I'm not sure.
Thank you for the answer, and I give the word back to the pro-rector.
Thank you very much, and it was indeed a short answer which concludes this official defense. Mr. Rombaut, dear candidate, the time appointed for defending your thesis has now officially passed. The degree committee will now withdraw to discuss the quality of your thesis and in particular the way you have defended your thesis, including the presentation you have given before. I request that you and your company await the results of our deliberation and our return to this aula. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Mr. Rombaut, dear candidate, I'm still calling you candidate because you didn't know what I'm going to tell you. The degree committee here present has assessed not so much the quality of your thesis, that has been approved, but particularly the way you have defended this thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee here has decided to grant you officially the degree of doctor. Professor van Mierlo is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor. Professor van Mierlo.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Ben Rombaut, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
If there's a photographer, he can come forward. Who makes the pictures? That's the lady. Okay, just one moment. The lady has to come. I give the word to Professor Van den Hoven, who is going to give the so-called MENS certificate.
Thank you very much. Namens het onderzoeksinstituut MENS ben ik tevens gemachtigd om dit officiële certificaat uit te reiken. De eigenaar van dit certificaat heeft aangetoond een brede opleiding in academische vaardigheden te hebben ontvangen en relevante ervaring te hebben opgedaan die bijdraagt aan een succesvolle loopbaan als zelfstandig onderzoeker.
You can make another picture. And there will be soon coming a third picture. So now I give the word to Professor Brone, who is going to give the so-called Euron certificate.
Namens Euron, de European Graduate School of Neuroscience, ben ik tevens gemachtigd om u het Euron-certificaat uit te reiken. Euron is een netwerk van 8 universiteiten in Nederland, België, Duitsland, Frankrijk en Luxemburg. Het certificaat wordt toegekend aan gepromoveerde onderzoekers die deelnamen aan meerdere internationale cursussen en conferenties van het Euron-programma, die aan een buitenlandse onderzoeksinstelling hebben gewerkt en die hun werk hebben gepubliceerd in internationale wetenschappelijke tijdschriften.
We will now continue with the laudatios, and the first laudatio will be given by Professor Van den Hoven.
Thank you very much, Mr. Prorector. Perfisia Ben. Voordat Tim het serieuze gedeelte van deze laudatio verzorgt, even wat luchtig vermaak van mijn kant. To meme or not to meme? That is the question. Amai Ben, hoe heb je dit klaargekregen? Meerdere losgeslagen promotoren die als compleet onafhankelijke richtingaanwijzers alle jouw kompas richting de eindstreep dachten te vormen. Met het angstzweet op Bens voorhoofd leefde hij iedere donderdag toe naar ons wekelijks overleg. Of zeg maar liever beleg. Wie komt er opdraven? Wie niet? Het zal toch niet, hè? Nee. Daniel had toch onderwijs deze week niet? Tim is op congres, toch? Bert heeft hoofdelijk slechte wifi. Soms ontsprong je de dans, maar soms ook niet. Hoi, sorry, ik kom net van onderwijs. Sorry, ik had me bijna vergist in de tijd. Het is ook 3 uur 's nachts hier. Hé jongens, nieuwe wifi. Nee. Mijn hele promotieteam is er. Dit overleef ik niet. Ines, help. Wat een chaos. Iedereen met zijn eigen advies, behalve het goede. Van links naar rechts. Ben werd gevraagd meer focus aan te brengen in zijn werk. En het enige dat wij hier allemaal deden, was iedere keer 1001 nieuwe ideeën aandragen. Het dreef Ben geregeld tot wanhoop. En Ben maar denken, hoe geraak ik hier nou weer doorheen? Het antwoord? Humor. Die van zijn begeleiders vooral negeren en zeker niet thuis nadoen. Want daar krijg je jongen van. Te laat. Nee, goede humor. Memes. Ja, want een echte memekoning Ben, dat ben je. Je hebt jezelf en ons hele lab zowat door iedere crisis geloosd met je memes. Corona, niks vaccineren, memes. Casuïstiek op de werkvloer, memes. Tegenvallende data, memes. Het niet meer zien zitten met je PhD-project, memes. Het is eigenlijk heel simpel. Een Ben zonder memes is als een thesis zonder paginanummers. Oeps. Ik moet opschieten, anders wordt de prorector zenuwachtig. Over de statistiek hebben we het zo meteen wel tijdens de receptie. Ik geef het woord snel door aan Tim, maar niet voordat ik je van harte heb gefeliciteerd. Je hebt het geflikt, Ben. Chapeau. Ik wens je alle goed voor de toekomst. Until we meet again.
Een grotere FVO-grant aan de achterkant. Ben, Dr. Rombaut, ik ben vereerd dat ik jou mag feliciteren met het behalen van je doctoraat. Graag wil ik er ook je ouders, je familie, je vrouw, je dochter mee in betrekken in die felicitaties. Want een doctoraat behalen doe je niet alleen, dat doe je als team. Misschien een kleine chroniek over de totstandkoming van je thesis waar we vandaag zijn geraakt. Mijn eerste kennismaking met jou dateert van 2017. Ik denk de cursus Screening, Diagnose en Therapie, waarin jij een werkstuk schreef samen met je groep rond de ziekte van Huntington. Je presenteerde je daar snel als de trekker en de denker samen met je BFF Kenneth om hier dit werkstuk tot stand te laten komen. Na een kort gesprek, na een van die coachingsessies, kwamen we wel snel tot het idee dat er een mogelijke stage wel een optie zou kunnen zijn. Want een denker en een trekker konden we zeker gebruiken in ons toen recent gestart onderzoek rond CRISPR. En dit hebben we ook zo opgezet samen met die paranimf, toen nog PhD-student Melissa en senior-student Essia. En je bracht het vrij goed tot stand. Hè. Dus na die 10 weken stage, het was wel een beetje een hurdle om tot die stage te komen, want er was natuurlijk een stagebegeleider die overtuigd moest worden dat jij dit project zou mogen doen. En daar moesten we dan ook een goede motivatietekst voor schrijven en dat is dat ook gelukt en je bent dus ook aan ons gelinkt voor die stage. Waarvoor dank ook aan de coördinator. Na afsluiting van die stage heb ik een teambarbecue georganiseerd. Ik weet niet of het barbecue of stoofvlees of iets anders was, maar ik herinner me dat jij tot de laatste man bent gebleven op die barbecue. En dat je een beetje aarzelend naar me toe kwam en zei wel, zo'n PhD, ik zou daar eigenlijk ook wel heel hard voor willen gaan. Zijn daar opties? Dan heb ik gezegd, ja, zeker kunnen we zeker bekijken. En ik denk dat het goed is dat we ook op een ander moment verder doorgesproken hebben, want ik herinner me dat je je auto hebt laten staan toen en je hebt laten ophalen om naar huis te gaan. Dus ik heb daarna ook nog een keertje geverifieerd of je dat echt wel wilde doen. Maar dat bleek het geval. We maakten toen een inhoudelijk plan op, en samen met Bert, met professor Bronen en met Sophie Kessels hebben we een seniorstage uitgedacht om uiteindelijk de verschillende verhaallijnen samen te laten komen, zoals we het ook in ons hoofd hadden, rond DISC en PDE en Alzheimer. Je hebt dan ook een grant daarop geschreven, een FVO-grant. Maar tijdens je seniorstage bleek al dat je een andere financieringsbron had gekregen, mede doordat je 2 paranimfen ook een FVO-grant behaalden. En je kon dus eigenlijk vrij relaxed naar dat PhD toeleven. Zelfs zo relaxed, en ik ga even citeren, dat je paranimfen me kwamen zeggen: we hebben Ben net verteld dat hij een FVO-grant heeft gehaald. Hij viel een beetje uit de lucht, maar was wel heel erg blij. Goed, de rest is geschiedenis, Ben. Gedurende de laatste 5 jaar heb je gewerkt aan de totstandkoming van deze thesis. Je start ook met het schrijven van een sterke review. Ik denk dat daar ook de termen wollig en climax of cliffhanger aan het einde van de zin voor het eerst gevallen zijn. Dat is intussen alweer een stuk beter. En ik denk dat je daar ook een grote stap hebt gezet. Dan kom ik tot volgende grant waar je aan meewerkt, een grotere FVO-grant, een high-risk, high-gain project. En ik denk dat het heel belangrijk is. Je hebt een heel grote bijdrage in het binnenhalen van die grant. En je hebt jezelf daar ook in verzekerd. En de support van 3 postdocs, die ook elk een fantastische bijdrage aan dit project hebben gehad. En ook dus tot totstandkoming van deze thesis. De memes zijn al benoemd. En ik denk de memes zijn legendarisch. Net zoals de titels van vele proefschriften in ons team waar jij in handen had, als ook het logo draagt jouw signatuur. De one-on-one meetings zijn ook al uitvoerig behandeld. Dus daar ga ik ook niet te veel dieper op ingaan. We moesten altijd maken dat we een uur inplannen, zodat we tot de essentie konden komen, of dat SIA kon wachten. Een van deze 2 opties. Ik denk ook aan de cursussen die we samen hebben gevolgd en de memorabele introducties met ik ben Tim en ik Tim ben. En toen het helemaal verwarrend werd, toen er een student kwam die zei ik ben Rombaut. Ik denk ook aan de PDE-meeting, de Gordon-meeting in Zwitserland, waar je met heel veel enthousiasme en omgekeerd evenredig veel rugpijn naartoe bent gegaan. Met de auto, zonder poster, met de kabouter van Bert, die we daar ook achter hebben gelaten. Ik denk dat we kunnen zeggen dat je het fantastisch hebt gedaan, je PhD-project, maar ook dat het geen pad over rozen was. Het was geen walk in the park. Je hebt het niet cadeau gekregen. Ik denk dat je ook vooral geleerd hebt wat je goed kan, wat je graag doet, ook wat je niet graag doet. En ik hoop dat je die inzichten gebruikt, ook in je toekomstige carrière. Ik wens je heel veel succes, Ben. Dan geef ik nu het woord terug aan de projector.
Dank u wel. Nou, dan hebben we al een hele laudatie gehoord. Dus ik wil eerst pas u, de nieuwe, wat wij de nieuwe jonge dokter Rombaut. Voelt goed? Heel goed, perfect. Ik wil u graag feliciteren namens de Universiteit Maastricht. I will switch to English because maybe we have some English guests here in the audience. So I would like to congratulate you on behalf of Maastricht University. In my congratulations, of course, is also your promotion team, who brought you in the last 5 years. And I heard with some downs and some ups, but I like to look to the ups mostly. And I said, it looks a good job. Indeed, I noticed when I received your book, I was puzzled because I noticed there are 2 books. And I thought, well, I missed something here because this is the book also without any page numbers. But that's fine with me. Okay. So I welcomed already and I thanked already your members of your promotion team. I also would like to thank the members of the Corona who came this morning, the 6 members of the Corona, and who asked you questions. It is much appreciated and is important, and we were pleased to see that you could answer all the questions. And I also can tell you that as the promotion team, the members of the Corona decided this morning to give you a unanimous positive decision, so there was not one negative point. That's a good point. In my congratulations, I also would like to add your family. They are hiding a little bit on the 4th lane. You could sit in the front because you are very important, so you are VIP. And I would like to say he did a great job. And by that, I'm going to conclude. And I said, this is how we're going to proceed. So basically, first of all, I'm going to conclude this session, use my hammer, as you can see over here. And how to proceed, we're going to make some pictures over here. And first we make a picture here in the aula, together with the online members who will appear on the screen. So I would like to ask the photographer to stay here. Then we're going to make a picture on the stairs. And then we were going to join you in the rafter. And the people, the audience here, do not have to wait for us. They can start already with the coffee, the tea, the pie, and even the beer, which is available, even in the Netherlands in the morning. And we will join you in about 10 minutes. Thank you very much.